Hello friends, welcome to BMH Learning. Here we are going to talk about restriction enzymes. Before we discuss about how restriction enzymes are useful in molecular biology, we must know what are these enzymes and how they operate. To get into this, let's suppose we have a bacteria over there and it has its genetic material. Then there is a bacteriophage or a virus which feeds on bacteria. So this virus infects the bacteria and injects fast DNA into the bacterium. As the viral DNA comes into the bacterium, it is sensed by, you can say, the immune system of the bacteria, which is nothing else but restriction enzyme itself. So these enzymes bind to the fast DNA, and we see fast DNA is cleaved and inactivated. This way the bacteria is protected from bacteriophage infection. This phenomenon of not allowing the growth of the virus is called host control restriction. Because the enzyme restricts the DNA replication, we call it as restriction enzyme. Now, there can be a question in your mind. What about the personal DNA of bacteria? Will it be a victim to its ancestor, that is, restriction enzyme? Well, no, it's because the bacteria is very clever. With the help of the enzyme methylase, the bacteria methylates the recognition sequences of restriction enzyme present on the bacteria. This way, the bacteria blinds and fools the enzyme, and the result is that it cannot bind to its recognition sequences, and the DNA is saved from getting cleaved. Now let's know the main function of these enzymes, that is, how they cleave their substrate and how we use them in gene cloning. Suppose we have this stretch of DNA containing the target gene to be cloned. The central feature of a restriction enzyme is that it has a particular recognition sequence at which it cuts a DNA molecule. Here we are taking an example of EcoR1. So what this enzyme will do is that basically it would scan all over the DNA until it finds this specific sequence, and whenever this particular sequence is found in the DNA, it would cleave the DNA at that point. So in the given stretch of DNA, the recognition sequence is highlighted. Apart from this, the arrow shows the cleavage sites. So the enzyme will cut the DNA from here by breaking the phosphodiester bonds present between these nucleotides. Now this is a plasmid which will be used to deliver the target gene. So first we have to introduce the gene of interest into this plasmid. For that to happen, we are going to open this plasmid by cutting it with the same enzyme used for cleaving the target gene. This results in the formation of overhangs, or technically saying sticky ends. If you notice clearly, these overhanging ends of the plasmid are complementary to the sticky ends of the target gene. This is what we want. Now the target gene can come and easily attach itself to the plasmid. This is how gene cloning is initiated. This was all. Thanks for watching.